Welcome to the Deep Dive, your essential shortcut to being well-informed. Today, we aren't just summarizing facts, we are exploring the foundational code of life.
We really are.
We're taking the most fundamental concepts in chemistry, atoms, bonds, water, and tracing their influence all the way up to the massive biological molecules that make you up.
Well, it's a pretty ambitious journey. If you're preparing for a major meeting, catching up on the field, or you're just driven by curiosity, this deep dive provides the absolute molecular logic for everything that happens in a living system.
It sounds like it.
We're moving from the microscopic rules of attraction all the way to the colossal structures of DNA and proteins.
Before we jump into the periodic table, I want to anchor all this science in a really powerful real-world story, the career of Dr. Kenneth Oldin. His work, I think, just perfectly demonstrates how understanding basic chemistry leads to life-changing biological breakthroughs.
It's a fantastic example. Dr. Oldin's story is really rooted in this social purpose.
Yeah.
He grew up in rural America, and he was really motivated by a desire to address health disparities, to help the poor. This mission, it just drove him through his studies in biology and chemistry, and that eventually led him to the National Cancer Institute.
And his early research there focused on a specific protein called fibronectin. What was so significant about this molecule?
Fibronectin is a glycoprotein, which means it's a protein that has these little sugar chains or carbohydrates attached to it.
Okay.
And researchers at the time had noticed that this protein was, it was consistently present on the outer surface of normal, healthy cells, but it was almost entirely absent on the surface of many cancer cells.
So the going hypothesis at the time was that the carbohydrates were some kind of necessary export tag.
Exactly. The idea was they were required for the cell to move the fibronectin out to its surface, and cancer cells, for some reason, had lost that ability.
Makes sense.
So to test this, Dr. Oldin and his colleague, Ken Yamada, needed a way to selectively stop those carbohydrates from being attached. In 1978, they used a newly discovered drug called tunicomycin, which does exactly that. It blocks the sugar attachment process.
And what did they find? I'm sensing a twist here.
Oh, it was a classic moment of scientific discovery, a huge surprise. They found that, completely contrary to the hypothesis, the protein was still exported just fine, even without the sugar chains.
So the tag idea was wrong.
Totally wrong. But, and this is the crucial part, the proteins that didn't have the carbohydrates were highly unstable. They just degraded really quickly. So the sugars weren't for export. They were essential for stabilizing the protein structure.
That is a fundamental reorientation of thinking.
Huge. It became one of the most highly cited papers of 1978 because it drove home the point we're going to keep revisiting today. Structure dictates function.
Right.
And even these minor chemical additions, like a little sugar chain, are absolutely vital for maintaining that functional structure.
And Dr. Oldin later took this molecular focus and applied it on a much larger scale, right? At the NIEHS.
He did.
Yeah.
As director of the National Institute of Environmental Health Sciences, he immediately broadened the scope of the whole institute.
Because historically, environmental health had focused pretty narrowly on what? Chemical carcinogenesis?
Precisely. The study of how specific chemicals cause cancer. Dr. Oldin expanded that mission to integrate social, behavioral, and genetic issues. He recognized that environment is so much broader than just chemical toxins.
So he was actually engaging with communities.
Actively, to identify their health concerns related to, say, disproportionate chemical exposure in different neighborhoods. It was a holistic approach.
And this approach also led him to launch the Environmental Genome Project.
Yes, which was incredibly forward-thinking at the time. The project used early genomic sequencing to figure out how an individual's own genetic makeup might make them more susceptible or maybe more resistant to certain environmental toxins.
It makes the research personal on a molecular level.
It does. And a major focus for him was children. He established these research centers dedicated to children's environmental health, recognizing that, infants and children are far more susceptible to toxins than adults are. Their bodies are still developing.
So his career is this perfect arc.
A perfect arc. From investigating how a tiny sugar stabilizes one protein to setting national public health agendas to protect entire vulnerable populations. And it all starts with chemistry.
That sets our mission beautifully. We're going to follow that arc. Our deep dive starts with the chemical context of life. Atoms, isotopes, and bonding. Then we'll spend some time on the unique properties of water. Next, we explore carbon, the molecular backbone of life, and its incredible versatility.
And then we'll bring it all together.
And finally, we will synthesize all of this by examining the four colossal classes of large biological molecules.
Okay, so to start, we have to acknowledge the absolute building blocks. Matter is just anything that takes up space and has mass. The purest forms of matter are elements, which are substances that you can't break down into other substances by chemical means. There are 92 of them that occur naturally.
And when these elements combine, that's where the magic really starts to happen. We get compounds.
Yes, compounds are two or more different elements combined in a fixed ratio. And this brings us right back to that first massive insight you hear about in biology, emergent properties.
The idea that the whole is greater than the sum of its parts.
It's the rule that governs everything. I mean, take sodium, Nan. It's a highly reactive metal. Then you have chlorine, Cl, which is a poisonous greenish gas.
Not things you'd want to interact with.
Definitely not.
Yeah.
But when they react chemically, the compound they form, sodium chloride, NaCl, is table salt. It's edible, it's stable, and it's required for life. The compound has characteristics, its emergent properties, that are completely distinct from its components.
Now, when we look at those 92 elements, the list of what's actually necessary for life shrinks pretty dramatically.
It really does. Only about 20 to 25% of them are considered essential for a healthy life and reproduction.
And what's amazing is that, life is chemically pretty simple at its core.
It is, overwhelmingly so. You're looking at oxygen, carbon, hydrogen, and nitrogen. That's it. That's like 96% of the mass of all living matter.
And then you have trace elements. These are needed in tiny, tiny quantities, but their absence can be catastrophic.
Iodine is the classic example. You absolutely need it for your thyroid gland to produce its hormones. If your diet lacks iodine, the thyroid gland tries to compensate by swelling up to this abnormal size.
That's a goiter.
That's a goiter. We only need about 0.15 milligrams a day, but that tiny chemical component makes the difference between basic health and severe endocrine problems.
So to understand how all these elements interact, we have to go inside the atom, the smallest unit that still has the properties of that element.
Right. The structure is, well, it's complex but beautiful. You have a dense, incredibly small atomic nucleus, and inside that you have positively charged protons and neutral neutrons. And orbiting that nucleus is the electron cloud, made up of these rapidly moving, negatively charged electrons. The opposite charges are what hold the whole system together.
So if you were in a lab trying to identify an element, what's the one number you'd look for?
The atomic number. It's defined by the unique number of protons in the nucleus. That number is the element.
And the mass number?
That's just a total sum of protons plus neutrons. This simple arithmetic is how we characterize matter. So let's take sodium again. Na-23, atomic number 11.
So that means 11 protons.
11 protons. And if it's a neutral atom, 11 electrons. And then 23 minus 11 gives you 12 neutrons. Simple as that.
But not all atoms of an element are identical, which brings us to isotopes.
Exactly. Isotopes are atoms of the same element that just differ in their number of neutrons, which changes their mass. Carbon-12 and carbon-13 are stable isotopes. Their nuclei stay intact forever.
But carbon-14 is different.
Carbon-14 is a radioactive isotope. Its nucleus is unstable, so it spontaneously decays, releasing particles and energy.
And that instability is exactly what makes radioactive isotopes so incredibly useful. It turns a decay process into a powerful scientific tool.
A very powerful tool. First, in medicine, we use them as radioactive tracers. A cell treats a radioactive atom exactly like its stable counterpart. So researchers can create a biologically active molecule, like glucose, but tag it with a radioactive isotope.
And you can follow it through the body?
You can track its path and see where it accumulates using detectors.
This is how PET scans work, right?
Exactly. In a PET scan, doctors inject radioactively labeled glucose. Tissues that are highly active, like tumors or certain parts of the brain, are metabolically hungry, so they gobble up that glucose really fast. By detecting the radiation, the scan creates this dynamic image showing where that highly active tissue is.
So it's invaluable for cancer monitoring.
And for diagnosing neurological disorders.
Yeah.
But beyond medicine, that fixed decay rate gives us our most reliable historical calendar. Radiometric dating.
This is fundamental to geology and paleontology.
The whole thing works because of this concept called half-life.
Okay, so that's the rate of decay, basically.
Exactly. It's the time it takes for exactly half of the parent isotope to decay into a stable daughter isotope.
Yeah.
And that rate, it's constant. It's totally fixed, like a clock.
So for archaeological work, carbon-14 is the famous one. How does that work, and what are its limits?
So C14 has a relatively short half-life, about 5,730 years, and it decays into nitrogen-14. When an organism is alive, it's constantly taking in carbon, so it maintains the same C14 to C12 ratio as the atmosphere.
But once it dies,
It stops taking in carbon, and that C14 clock starts ticking. The C14 begins to decay exponentially, so we just measure what's left and calculate how many half-lives have passed.
but because that half-life is so short, it has a time limit.
It does. After about, say, 75,000 years, there's just too little C14 left to measure reliably. For dating rocks or fossils that are millions of years old, scientists use different isotopes with much, much longer half-lives, like potassium-40. But the principle is exactly the same. Hashtag tag 1.2 electron energy and chemical reactivity. So while the nucleus gives an atom its identity and mass, it's the electrons that govern all the chemical interactions.
And the behavior of those electrons is all about energy, specifically potential energy.
Right. Energy is just the capacity to cause change. And potential energy is the energy that matter has because of its location or its structure.
So for an atom, that means an electron's potential energy is based on its distance from the nucleus.
The farther away it is from that positive nucleus, the more potential energy it has.
I always found the analogy of a staircase really helpful here.
It's a perfect analogy. An electron can't exist in the space between energy levels, just like a ball can't rest between steps on a staircase. They only exist at these fixed energy levels, which we call electron shells.
So to move an electron up to a higher shell, farther from the nucleus, the atom has to absorb energy.
Like lifting the ball up the stairs. And conversely, when an electron falls back down to a lower shell, it releases that energy, often as light or heat.
And it's the electrons in the outermost shell that are the most important for chemistry.
Yes, that's the valence shell. And the electrons in it are the valence electrons. These are the ones that dictate whether an atom will react and how it's going to bond.
Because atoms want to have a full valence shell. That's their most stable state.
Exactly. They seek the lowest possible state of potential energy, which is achieved when that valence shell is complete. And if it's already complete, we call those the inert elements.
The noble gases, like neon or argon.
Right. They're chemically unreactive because their valence shells are full. Every other atom with an incomplete valence shell is reactive. It's going to try to interact with other atoms to complete that shell, either by sharing or by transferring electrons.
So we move beyond these 2D shells to think about the 3D space they occupy, which are orbitals.
An orbital defines the three-dimensional region where you're likely to find an electron 90% of the time. The first shell has one spherical s orbital. The second shell has one spherical s orbital and three dumbbell-shaped p orbitals.
And each one of those orbitals can hold a maximum of 2 electrons.
A maximum of 2. And this specific structural arrangement is what determines the geometry of the bonds that they'll eventually form.
So that drive to complete the valence shell is the entire engine of chemical bonding. And when atoms share electrons to get stable, we get the covalent bond.
Which is the strongest type of bond in most biological molecules. And the number of bonds an atom can form, its valence, is just determined by how many unpaired electrons it has.
Right, so we have to remember the key valences. Hydrogen is 1, oxygen is 2, nitrogen is 3.
And carbon is 4. These numbers dictate the entire molecular architecture of life.
So in a molecule like methane, CH4, carbon shares 1 pair of electrons with each of 4 hydrogen atoms. That gives you 4 single covalent bonds, and everybody's happy.
Everybody's valence shell is full.
Exactly.
Yeah.
But now we have to introduce a concept that complicates that sharing, polarity. And this is all determined by electronegativity.
Which is just how strongly an atom pulls on the electrons it's sharing.
It's the atom's attraction for those shared electrons. If the 2 atoms are the same, say 2 hydrogens forming H2, they pull equally. The sharing is perfect.
And that's a nonpolar covalent bond.
But if they're different, especially if one atom is highly electronegative and oxygen is the classic bully here, the sharing becomes unequal.
And that creates a polar covalent bond.
Yes, where the shared electrons spend more time orbiting the highly electronegative atom, this unequal distribution is vital. It creates these partial charges, delta minus and delta plus, across the molecule.
And the water molecule, H2O, is the ultimate example.
Absolutely. Oxygen is greedy. It pulls the electrons away from the hydrogens, so the oxygen side becomes partially negative, and the hydrogen sides become partially positive. This polarity is the single feature that makes life on Earth possible. But sometimes that electronegativity difference is so extreme that sharing is just abandoned completely.
And that's where you get an ionic bond. The highly electronegative atom just outright strips an electron from its less electronegative partner. This transfer creates ions.
A positive cation and a negative anion.
Right. The atom that lost the electron becomes a positive adhesion, and the one that gained it becomes a negative anion. The ionic bond is just the very strong electrostatic attraction between those opposite charges.
And the resulting compounds are salts, like NaCl.
And this is where the environmental context matters so much. In a dry salt crystal, those ionic bonds are incredibly strong, holding this rigid lattice structure.
Dissolve it in water.
And the whole thing changes. The water molecules surround and shield the ions, which dramatically weakens that attraction.
And this has huge practical consequences in medicine.
Huge. Many drugs are made of salts because they're stable in their dry, packaged form. But as soon as they hit the watery environment of your body, they quickly dissociate, releasing the active ions to go and interact with their biological targets. The environment determines the bond strength, #tag 1.4 weak interactions, shape, and chemical reactions.
Okay, so we've covered the strong bonds. Now let's move to the weak interactions. These are temporary, individually fragile, but absolutely essential for complex biology.
They're like the molecular Velcro of life. And the most important one is the hydrogen bond.
Right.
This happens when a partially positive hydrogen atom that's already in a polar covalent bond with an oxygen or a nitrogen is then attracted to another nearby electronegative atom.
This is the bond that links water molecules together, and it's also crucial for stabilizing the giant 3D structures of proteins and DNA.
And the second type is the van der Waals interaction. Now, this one occurs even between nonpolar molecules.
How does that work if there are no charges?
Well, it's because electrons are always moving. So at any given instant, the electrons in a molecule might just randomly cluster on one side. This creates a transient, a momentary positive and negative region.
So even a non-polar molecule has these tiny, fleeting dipoles.
Constantly. And these momentary positive and negative spots cause these very slight, very weak attractions between adjacent molecules. They are the weakest of the interactions, but they're highly cumulative.
And the ultimate example of that cumulative power is the gecko.
It's the perfect example. A gecko's toes are covered in millions of microscopic hairs that maximize the surface area with whatever it's climbing. There's no glue, no suction. The entire ability of that gecko to walk straight up a sheet of glass is due to the astronomical cumulative effect of millions and millions of these incredibly weak van der Waals interactions.
Wow. So whether it's strong bonds or weak interactions, they all dictate molecular shape, and we can't overstate this. Molecular shape dictates biological function.
Shape is absolutely everything. Remember how carbon forms 4 single bonds? That results in a very specific tetrahedral geometry with angles of 109.5 degrees.
And water is V-shaped.
A V-shape of 104.5 degrees. This specific geometry ensures that molecules can recognize and bind to each other like a lock and a key.
This specificity is what allows for molecular mimicry, and the classic example involves our body's own pain receptors.
Think about the drug morphine. Why does it work as a painkiller? It's because the specific 3D structure of the morphine molecule is strikingly similar to the shape of our brain's natural pain-relieving molecules called endorphins.
So they both fit into the same receptor protein.
They both fit perfectly. The cell essentially gets the same no pain signal whether the molecule is an endorphin from inside your body or morphine from outside. The shape is the message. If morphine were just slightly misshapen, it would bind and it would be useless.
Finally, these bonds are constantly being made and broken through chemical reactions.
Right, which are just processes that change the chemical composition of matter. The most essential reaction for life is photosynthesis, where plants rearrange carbon dioxide and water into the high-energy sugar glucose and release oxygen.
And the key principle there is that matter is conserved. The atoms are just rearranged.
Never created or destroyed, and these reactions are generally reversible. When the rate of the forward reaction equals the rate of the reverse reaction, we reach chemical equilibrium.
And it's important to remember this is a dynamic state, not a static one.
Very dynamic. Reactions are still happening in both directions, but the concentrations of the reactants and products have stabilized. There's no net change over time.
Okay, so we've established that the water molecule, H2O, is V-shaped and highly polar because of oxygen's high electronegativity. And that polarity is the starting point for everything that follows.
It is. That polarity means the partial positive hydrogens of one water molecule are constantly attracted to the partial negative oxygen of its neighbor, forming hydrogen bonds.
And in liquid water, these bonds are constantly breaking and reforming.
At a blistering pace. They're extremely fragile, but there are so many of them. And it's the collective force of all these shifting hydrogen bonds that gives rise to the four emergent properties that make water just uniquely suited for life.
Let's start with the dual action of cohesion and adhesion.
Cohesion is just water molecules sticking to themselves because of those hydrogen bonds. This is what creates high surface tension.
Right, you see that when water forms a dome on a penny or when a spider can walk on a pond.
Exactly. And cohesion works hand in hand with adhesion, which is water clinging to other substances, as long as those substances are also polar or ionic.
And their synergy is critical for plants.
It's how a giant redwood gets water to its top leaves. Water evaporates from the leaves, and that pulls the entire column of water up through the plant's vessels. That upward pull against gravity is maintained by cohesion, the water molecules sticking together, and adhesion, the water clinging to the walls of the vessels.
The second major property is water's ability to moderate temperature.
Right, and for this, we have to be specific with our terms. Kinetic energy is energy of motion. Thermal energy is the total kinetic energy of all the molecules in a substance. And temperature is the average kinetic energy.
And heat is measured in calories.
Yes, the heat required to raise 1 gram of water by 1 degree Celsius.
Now, water has a remarkably high specific heat. Why does it take so much energy to heat water up?
It's those hydrogen bonds again. Specific heat is the amount of energy you have to absorb for 1 gram of a substance to change its temperature by 1 degree Celsius. For water, that value is high because a lot of the initial heat energy goes into disrupting that massive network of hydrogen bonds.
So you have to break the bonds first before you can even speed up the molecules.
Exactly. Only after those bonds are broken can the temperature actually rise. Those hydrogen bonds act like a thermal sink.
And this has huge consequences.
Global and local. Large bodies of water absorb massive amounts of solar heat, stabilizing ocean temperatures and moderating coastal climates. And internally, because your body is mostly water, it allows you to maintain a stable internal temperature.
And the second part of this is water's high heat of vaporization.
Right. To turn liquid water into a gas, to evaporate it, you have to break every single one of those hydrogen bonds. That takes a huge input of energy.
And the consequence is evaporative cooling.
Yes. When water evaporates, it's the molecules with the greatest kinetic energy, the hottest, fastest ones, that are able to escape the liquid surface.
And when the fastest molecules leave, the average temperature of what's left behind drops.
This is the critical mechanism behind sweating in humans or panting in dogs. It's an incredibly efficient way to dissipate large amounts of heat and prevent overheating. Hashtag tag 2.2 ice floating, and water as the solvent of life.
The third emergent property is maybe the most counterintuitive. Water is less dense as a solid than as a liquid. Why does ice float?
It is the hydrogen bonds, one last time, at work. As liquid water cools below 4 degrees Celsius, the molecules slow down. At 0 degrees, the hydrogen bonds become fixed. They lock the water molecules into this stable crystalline lattice structure.
And that lattice structure holds the molecules farther apart than they are in the liquid.
Exactly. The lattice is more ordered but less dense. Ice is about 10% less dense than liquid water at 4 degrees Celsius.
And if ice sank, as most solids do,
Lakes and oceans would freeze solid from the bottom up, and aquatic life would be impossible. Because ice floats, it forms this insulating layer on the surface, protecting the liquid water and all the organisms underneath it from freezing.
but this same phenomena is now a major concern with climate change.
It is. The floating sea ice in the Arctic provides essential habitat for keystone species like seals and polar bears. The rapid loss of that ice is a catastrophic loss of habitat, and it shows just how crucial this one chemical property is to entire ecosystems.
The fourth property is water's role as the universal solvent of life.
It's an unusually versatile solvent because of its high polarity. When you dissolve an ionic compound like salt in water, the partially negative oxygen atoms are attracted to the positive sodium ions.
And the partial positive hydrogens are attracted to the negative chloride ions.
Right. Water molecules surround each ion, forming a protective hydration shell that pulls them away from the salt crystal.
And this defines what does and doesn't dissolve in a biological system.
Any substance that is ionic or polar like sugars or proteins will readily interact with water. We call those hydrophilic or water-loving.
And substances that are nonpolar, like fats and oils.
They repel water. They're hydrophobic or water-fearing. In a watery environment, hydrophobic molecules will all clump together to minimize their contact with the water.
To manage these solutions in a lab, we need standardized units, which brings us to the mole.
Right, atoms are too small to count, so we use the mole as a counting unit. It's Avogadro's number, 6.02 times 10 to the 23rd molecules.
And the beauty of the mole is that a mole of anything, in grams, has the exact same number of molecules as a mole of anything else.
This standardization is critical, and it leads to molarity, which is the gold standard for concentration in biology. Molarity is just the number of moles of solute per liter of solution. It allows for reproducible science everywhere. Hashtag 2.3 acids, bases, and pH control.
Okay, so even in pure water, a tiny fraction of molecules are constantly dissociating.
It's a very small number, but it's a crucial process. A water molecule will occasionally break apart into a hydrogen ion, H plus on, an hydroxide ion, OH.
And in pure water, those ions are in perfect balance.
Perfect balance. The concentration of both is 10 to the minus 7 molar. But introducing acids or bases dramatically shifts that equilibrium.
An acid increases the hydrogen ion concentration.
Right, it donates H plus anina. A base does the opposite. It reduces the H plus concentration, either by accepting an H plus or by releasing OH ions, which then combine with H plus on.
And we use the pH scale to quantify this.
Yes, where pH equals the negative log of the hydrogen ion concentration. And because it's a logarithmic scale, a drop from pH 7 to pH 6 is a tenfold increase in acidity. That's why even small pH changes can have huge chemical consequences.
And biological fluids need to be kept in a very narrow pH range.
Generally between pH 6 and 8. And maintaining that delicate balance is the job of buffers.
So buffers resist those changes.
They're chemical systems, usually a weak acid-base pair, that resist sudden changes in pH. If H plus starts to rise, the buffer accepts the excess. If H plus starts to drop, the buffer donates H plus Fen.
What's the most critical buffer in the human body?
That would be the carbonic acid bicarbonate buffer system in your blood. It keeps your blood pH tightly controlled right around 7.4. If it deviates much from that, it could be fatal because your proteins and enzymes lose their functional shape.
Speaking of pH stability, we have to talk about ocean acidification, a global threat from a simple chemical reaction.
It's a direct consequence of rising atmospheric CO2. About a quarter of that CO2 dissolves into the oceans. Once it's there, it reacts with water to form carbonic acid, which immediately lowers the ocean's pH.
But the real problem isn't just the acidity, it's the loss of a key building block.
Exactly. The increased concentration of H plus ions combines with the available carbonate ion, CO3, 2-, basically stealing them. And those carbonate ions are absolutely essential for marine organisms like corals and mussels to build their shells and skeletons out of calcium carbonate.
So by lowering the pH, we are chemically starving these shell-building organisms.
We are, and we're threatening the entire base of the marine food web. It's a profound example of how a small chemical shift can have a monumental biological impact. So if water is the medium of life, carbon is the backbone. Organic chemistry, the study of carbon compounds, is essentially the chemistry of life itself. All the incredible complexity and diversity of organisms flows from carbon's unique bonding capacity.
And that raises the question, how did these complex organic molecules first arise on early Earth? The classic experiment here is from Stanley Miller in 1953.
Miller's experiment was a landmark attempt to show abiotic synthesis-making organic molecules from inorganic stuff under primitive Earth conditions.
Right, he set up this apparatus that simulated ancient oceans and an ancient atmosphere with sparks to mimic lightning.
And he succeeded spectacularly. After just a week, the water in his apparatus contained a whole variety of complex organic compounds, including amino acids.
The building blocks of proteins.
Yes. It was strong support for the hypothesis that the basic organic chemistry for life could have arisen spontaneously.
Now, we think the ancient atmosphere might have been a bit different than what he modeled.
That's true, but later experiments using these updated gas compositions still produced organic molecules. The underlying conclusion is still robust. The chemistry of carbon is so versatile that organic building blocks will naturally form under a wide variety of plausible early Earth conditions.
So what gives carbon this unrivaled versatility?
It really boils down to its valence. Carbon has 4 valence electrons, so it has to form 4 stable covalent bonds to complete its shell.
That makes it a perfect intersection point.
A perfect intersection. It can branch out in 4 directions at once, forming chains, rings, and complex 3-dimensional scaffolds.
And these bonds dictate the shape of the molecule.
If carbon forms 4 single bonds, you get a tetrahedron shape. But if it forms a double bond, the molecule becomes flat, or planar. And that shift in geometry dramatically affects how the molecule can interact with other things.
These linked carbon atoms form the carbon skeleton, the backbone of all organic molecules.
And these skeletons can vary immensely in length, branching, double bond position, and they can even form rings. This endless variety in the skeleton alone creates huge structural diversity.
And if the skeleton is just carbon and hydrogen, we call it a hydrocarbon.
Hydrocarbons are critical because the CH bonds are nonpolar, which makes them hydrophobic. They don't mix with water. And they are very energy rich. The long hydrocarbon tails of fat molecules are the most efficient form of energy storage for animals.
Okay, so moving from the skeleton, we see that complexity can arise even when the chemical formula is identical. And that's thanks to isomers.
Right, isomers have the same molecular formula but different structures and therefore different properties.
We categorize them into three types. The first is structural isomers, which just differ in the covalent arrangement of their atoms.
Like a straight chain versus a branch chain.
Exactly. The second type, cis-trans isomers, highlights the critical role of geometry around an inflexible double bond.
If two identical groups are on the same side of the double bond, it's the cis isomer. If they're on opposite sides, it's the trans isomer. This shape difference is crucial. The harmful effects of trans fats are due to their straight, packed structure, which is chemically different from the natural kinked structure of cis fats.
And the third type, enantiomers, are arguably the most important biologically. They're mirror images of each other.
Non-superimposable mirror images, like your left and right hand. They have the same components, but they are not identical. In biological systems, this handedness is incredibly specific, and usually only one of the enantiomers is active.
And this has huge consequences in pharmacology.
Massive. Take ibuprofen. It's sold as a mix of both enantiomers, but only the S enantiomer actually works as a pain reliever. The other one is inert.
Wow.
In the case of the asthma drug albuterol, one enantiomer is therapeutic, but the other one actually counteracts its effect. It just shows that molecular architecture is absolutely not negotiable in drug design.
So while the carbon skeleton provides the basic scaffold, the actual chemical behavior, the reactivity, the solubility is determined by the functional groups attached to it.
Functional groups are these small clusters of atoms that are almost always involved in chemical reactions. They give the molecule its specific properties. We focus on 6 highly reactive groups that are also hydrophilic, meaning they make the molecule more soluble in water.
Let's focus on the two that are key players in controlling pH and charge.
That would be the carboxyl group and the amino group. The carboxyl group, COOH, acts as an acid. It can donate an H+ ion, leaving the group negatively charged.
And the amino group does the opposite.
The amino group, NH2, acts as a base. It can pick up an H+ from the solution, becoming positively charged. These two groups are vital because they allow amino acids and proteins to be charged at cellular pH.
And the sulfhydryl group plays a unique structural role.
Yes, the sulfhydryl group, SH, is found in the amino acid cysteine. Two of these can react together, forming a strong covalent link called a disulfide bridge. These bridges are like molecular rivets that help stabilize the complex 3D folding of many proteins.
And we can see the combined impact of functional groups when we compare the sex hormones, estradiol and testosterone.
They're both steroids built on the same 4-ring carbon skeleton. But the addition of just a few different functional groups results in massive differences in their shape and function, which in turn dictate the development of male and female sexual characteristics.
Finally, the most important molecule for cellular energy, ATP, is defined by its functional groups.
Adenosine triphosphate. It's adenosine attached to 3 phosphate groups. ATP isn't a long-term storage molecule like fat. It's the immediate energy currency of the cell.
And that energy is in the bonds between the phosphates.
Exactly. When the cell needs energy, water hydrolyzes that terminal phosphate bond, releasing a burst of energy that powers nearly all cellular work.
We now move to the pinnacle of biological organization, the large biological molecules, or macromolecules. These are the carbohydrates, proteins, and nucleic acids.
And the overarching structural principle here is the polymer concept. Most macromolecules are polymers, these long, chain-like molecules that are constructed from repeating smaller subunits called monomers.
With lipids being the one exception.
Lipids are the exception. They're large, but they don't form true chains of repeating monomers.
And building and breaking these huge chains relies on 2 standardized enzyme-driven reactions.
To build a polymer, the cell uses a dehydration reaction. This process covalently links 2 monomers by removing a molecule of water. That's synthesis.
And the reverse process is for breakdown.
That's hydrolysis, or water breakage. Hydrolysis breaks the covalent bond between mostas by adding a water molecule. This is fundamental to digestion, where you break down large food polymers into absorbable monomers.
What I find most remarkable is the sheer variety of life that's built from such a limited chemical toolkit.
This is the elegant molecular logic of life. All living things are built from only about 40 to 50 common monomers.
That's it.
That's it. 20 amino acids, a few sugars, 4 nucleotides. It's like using a 26-letter alphabet to write millions of unique books. The incredible diversity of life comes entirely from the unique sequence and arrangement of those few monomers. So let's begin with carbohydrates, which includes sugars and their polymers. The monomers are the monosaccharides, or simple sugars.
Like glucose, C6H12O6.
Right. And while we often draw them as linear chains, in the watery environment of a cell, they spontaneously form stable ring structures. They serve as the major fuel for cells and as raw material.
And when two monosaccharides join, we get a disaccharide, connected by a glycosidic linkage.
Key examples are sucrose, which is table sugar, and lactose, milk sugar. The inability to break the glycosidic linkage in lactose because you lack the lactase enzyme is what causes lactose intolerance.
But the real functional power of carbs comes with polysaccharides, polymers of hundreds or thousands of monosaccharides.
And they have two primary roles: energy storage and structural support. For storage, plants use starch, which is a polymer of alpha-glucose. It typically forms helical structures.
And animals use glycogen.
Glycogen is also a polymer of alpha-glucose, but it's much more heavily branched than starch. This branching provides many free ends from which glucose can be rapidly cleaved off when an animal needs a quick burst of energy.
For structure, the most abundant organic compound on Earth is cellulose.
Cellulose forms the tough structural component of plant cell walls. It's also a polymer of glucose, but here is the single most important structural difference. Cellulose uses the beta-glucose monomer,
Okay.
and in a cellulose chain, every beta-glucose is flipped-oriented upside down, relative to its neighbor.
And that tiny chemical flip completely changes the polymer's properties.
And completely, that flip forces the cellulose chain to be straight and unbranched. These straight chains can then align parallel to each other and form hydrogen bonds between them. This creates these incredibly strong cable-like microfibrils.
And this structural difference is why we can digest starch, but not cellulose.
Exactly. Our digestive enzymes are shaped to hydrolyze the alpha linkages in starch. They physically cannot engage with the beta linkages of cellulose, so cellulose just passes through us as insoluble fiber.
And there's another structural polysaccharide, chitin.
Chitin is used by insects and crustaceans to build their hard exoskeletons. It's similar to cellulose, but the glucose monomer has a nitrogen-containing group attached, which makes the material both strong and flexible. So lipids are defined not by a common structure, but by their shared trait. They're hydrophobic. Their chemistry is dominated by nonpolar CH bonds, so they mix poorly with water.
The most common type of lipid is the fat molecule, or triglyceride.
A fat is constructed by linking one glycerol molecule to 3 fatty acid molecules. Their main biological role is energy storage. They pack more than twice the energy per gram compared to carbohydrates.
And we classify fats based on the saturation of their fatty acid tails.
Right. Saturated fats have no carbon-carbon double bonds. Their hydrocarbon tails are straight, so they can pack together tightly. That's why animal fats, like butter, are solid at room temperature.
And unsaturated fats have double bonds.
One or more. And in nature, these double bonds almost always have a cis configuration, which introduces a rigid kink into the chain. This kink prevents tight packing, making them liquid at room temperature. These are the oils.
We mentioned that hydrogenation can create dangerous trans fats.
It does. It artificially converts some of those natural cis bonds into trans bonds. The trans configuration straightens the chain back out, allowing it to pack like a saturated fat, but this altered structure has been specifically linked to heart disease.
Next up, phospholipids, the foundation of all cell membranes.
Phospholipids are built with one glycerol, 2 fatty acid tails, and a hydrophilic phosphate group. This structure makes them amphipathic.
Meaning they have a water-loving head and water-fearing tails.
A hydrophilic charged head and 2 long hydrophobic tails.
Yeah.
And their amphipathic nature determines their unique behavior in water.
They spontaneously form a bilayer.
Yes, the hydrophobic tails cluster inward, shielding themselves from water, while the hydrophilic heads face outward, interacting with the watery environment inside and outside the cell. This bilayer is the crucial barrier that defines a cell.
And finally, the steroids.
Steroids are lipids, recognized by their carbon skeleton of 4 fused rings. Cholesterol is a key steroid. It maintains the fluidity of cell membranes and serves as the precursor from which your body makes crucial signaling molecules like the sex hormones. Proteins are the most structurally and functionally diverse macromolecules. They're involved in nearly every process, from movement and transport to defense and signaling.
And their most vital function is catalysis. Most enzymes, which regulate metabolism, are proteins.
Exactly. Enzymes dramatically speed up chemical reactions, and all proteins are polymers built from just 20 types of amino acid monomers.
And each amino acid has a central carbon, an amino group, a carboxyl group, and a variable R group.
And that R group, or side chain, is what gives each amino acid its unique chemical properties, nonpolar, polar, or electrically charged. These amino acids are linked by peptide bonds to form a polypeptide chain.
And the ultimate function of that protein is encoded entirely in its unique, precise, three-dimensional shape.
Yes, and we define this shape using 4 structural levels.
Mhm.
First, the primary structure is just a unique linear sequence of amino acids. It's the blueprint.
Second, the secondary structure.
These are local, repetitive coils called alpha helices and folds called beta-pleated sheets. And these are stabilized entirely by hydrogen bonds formed between the atoms of the repeating polypeptide backbone. The R groups are not involved here.
Third, the tertiary structure.
This is the overall intricate 3D shape of a single polypeptide chain. And this level is stabilized by interactions between the R groups.
So this includes hydrophobic interactions, ionic bonds,
And the very strong covalent to sulfide bridges between cysteine monomers.
and finally, quaternary structure.
This level only applies to proteins made of 2 or more polypeptide subunits that aggregate to form the final functional molecule. Hemoglobin, which carries oxygen in your blood, is a great example. It's an aggregate of 4 distinct polypeptide chains.
And this precise structure is so sensitive that a tiny error in the primary sequence can be devastating. This brings us back to sickle cell disease.
Sickle cell disease is a molecular disease caused by a single amino acid substitution. Valine, which is nonpolar, replaces glutamic acid, which is charged and hydrophilic, in the primary structure of hemoglobin.
And this one change introduces a hydrophobic patch on the surface of the protein.
And in low oxygen conditions, these hydrophobic patches on abnormal hemoglobin molecules stick to each other to avoid the water. This causes the molecules to aggregate into these long, rigid fibers.
Which deforms the red blood cell.
It deforms the flexible red blood cell into that characteristic sickle shape, which then clogs small blood vessels. It perfectly illustrates that a single amino acid substitution is enough to change the protein's overall shape and completely destroy its function.
So because shape is everything, if a protein loses its native shape, it's rendered inactive. That's denaturation.
Yes, denaturation happens when environmental conditions like pH or temperature are altered, destroying the weak interactions that stabilize the secondary and tertiary structure. And we now know that protein misfolding is linked to many serious neurodegenerative diseases like Alzheimer's and Parkinson's. So the information that dictates the primary structure of every single protein, the blueprint for all of life, is stored in our final class of macromolecules, nucleic acids.
We're talking about DNA and RNA, which define the gene as the unit of inheritance.
DNA stores the genetic information and dictates its own replication. It also dictates the synthesis of RNA. RNA then carries that message from the nucleus out to the ribosomes, where the protein is actually made.
So that's the central flow of information, DNA to RNA to protein.
Exactly. The monomers of nucleic acids are nucleotides. And each nucleotide has 3 components.
A phosphate group, a 5-carbon sugar, and a nitrogenous base.
Right. The bases come in 2 families. The single-ring pyrimidines, CT and U, and the double-ring purines A and G. And the sugar is also different. DNA uses deoxyribose, while RNA uses ribose.
These nucleotides link up to form a polynucleotide chain.
They're joined by phosphodiester linkages, creating a sugar-phosphate backbone that has directionality running from the 5' end to the 3' end. And the sequence of bases along this chain is where the genetic information is encoded.
And DNA famously exists as a double helix.
Yes, 2 polynucleotide strands winding around each other. The strands are anti-parallel. One runs 5' to 3', the other runs 3' to 5', and they're held together by hydrogen bonds between the paired bases in the interior.
And the pairing is rigidly complementary, which is the key to replication.
Adenine, A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. This precise, predictable pairing is the structural basis for how DNA can be copied identically every time a cell divides.
And the modern era of biology has been completely transformed by our ability to read this genetic code.
Through genomics, the massive sequencing of entire genomes, and proteomics, the large-scale analysis of proteins. This data allows scientists not only to track diseases, but also to trace evolutionary relationships back hundreds of millions of years. #tag outro. We have covered immense ground, moving from the rules of atomic attraction all the way to the informational complexity of the gene. To synthesize, I think you should remember three major takeaways.
First, structure is function, and all biological properties are emergent. The complexity we see is just an emergent consequence of simple rules of atomic structure and bonding, driven by carbon's versatility and electronegativity.
Second, water is the indispensable medium for life. Every single unique property of water, its cohesion, its ability to stabilize temperature, its floating solid form, its solvent action, is entirely dependent on the collective strength of its hydrogen bonds.
And third, the four classes of macromolecules, carbohydrates, lipids, proteins, and nucleic acids, are all built on that carbon scaffold, and their ability to do their jobs is dictated by their precise, specific three-dimensional shape.
Right. I mean, the insights from Dr. Olden's work to the catastrophe of sickle cell disease all demonstrate how acutely sensitive biological function is to molecular shape.
And that sensitivity brings us to our final thought. We've seen how altering basic chemistry like decreasing ocean pH can dramatically impair calcification at the base of the marine food chain. We also know that proteins require a precise pH to maintain their functional structure.
Hmm.
The chemical specificity that allows life to thrive is so finely tuned that we have to ask, what other subtle chemical balances, whether internal, like protein folding, or external, like atmospheric gas exchange, are currently sustaining the staggering diversity of life that we might unknowingly be pushing toward an irreversible chemical tipping point?
A question that connects every single element we discussed today, from the proton all the way to the planet.
Thank you for joining us on this essential deep dive into the chemistry that makes life possible. We'll see you next time.